Hello everyone, this is lecture 6 and in this lecture we will learn about enzyme catalytic mechanism. So we already know what catalysis is. Catalysis is a process that increases the rate at which a reaction approaches equilibrium, and in this process it lowers the free energy of activation by stabilizing the transition state. And the mechanism by which enzymes do that, they are of different types. Okay, so these are the major types by which enzyme exert their activities. Okay, so like acid base catalysis, covalent catalysis, metal ion catalysis, electrostatic catalysis, proximity and orientation effects, preferential binding of the transition state complex. So these are the types of catalytic mechanisms that enzymes employ. Okay, so now from the next slide I will talk about more details about these mechanisms. First, acid base catalysis. So in this kind of catalysis, the enzyme, they act as a acid. So what acids do? Uh, in general, acid catalysis is a process in which partial proton transfer from a Bronsted acid, okay, which lowers the free energy of a reaction's transition state. In the contrary, in general base catalysis, increases reaction rate by partial proton abstraction by Bronsted base. Okay. However, some reactions may be simultaneously subject to both process. Okay, that is a concerted general acid-base catalyzed reaction. So, in general, we can say that in this kind of catalysis, acid-base catalysis, the enzyme, they act like a acid or base. Okay, so if the enzyme, they act like a acid, that means they uh, donate a proton, then that it is called a acid catalysis. Or if the enzyme, it acts like a base, that is it abstract a proton, is called general base catalysis. So if you see the example, okay, so this reaction, keto to enol. Okay, keto is produced from enol. So if reaction uh, is if reaction occurs in an uncatalyzed way, so no catalysis, in that case what happens, to become enol from keto, this changes are necessary. What changes? If you look at the structure of keto and enol, keto is R, no, CRO CH2H, and enol is CO CH2R. That is this proton, it if we look at the structure, it looks like that this proton, it transferred to this oxygen. Okay, or we can say that this bond need to be broken down, so this hydrogen will get released and one hydrogen or proton will join with the oxygen, thus enol will be formed. Okay, so without any catalysis, this reaction rate will be very slow. But if there is catalysis such as enzymes, they can uh, increase the rate to a tremendous extent. Okay, so let's see how the enzymes help this transformation. Okay, so first we'll talk about the acid catalysis. So say this is an enzyme AH, and this enzyme, it acts it can act like a acid. That means it can donate a proton. So in that case, the acid will donate the proton to the keto. Okay, and this proton will bind with oxygen. Okay, and eventually this will facilitate the release of hydrogen from the keto structure and enol will be formed. Okay, so how this donation of proton by this enzyme facilitate the reaction or helps the reaction to occur faster? So when we know hydrogen is when this hydrogen get released from the acid, or they charged positively, because hydrogen ion, we know they lack electron. So if they want to bind with the oxygen of the keto, they need electron. That means the keto have to supply the electron to the proton. So how they do that? This bond, we know that the chemical bond, they are formed by electron. Okay, the two molecules, they share the electron and form the covalent bond. So as if this oxygen, they want to bind with the hydrogen ion, then that means with the proton, they need to supply oxygen, and this they need to supply electron, and this electron comes from this bond. Okay, so this double bond, they begin to break down, and the electron released from here are shared with the proton. So thus a bond between oxygen and hydrogen can form here. Okay, and because of this breakdown of the bond here, electron lacks in this place. So the electron which was shared in this bond, they tend to transfer from here to here. So that this bond, it breaks down and a double bond is formed here. Okay, so eventually the enol is formed. So in this way, the this enzyme, it helps the reaction to proceed. Okay, and as doing so, this enzyme, it acts as a acid. It is called acid catalysis. Okay. Okay, now what happened if the enzyme acts like a base? Okay, so if, so suppose this B, it's the enzyme, it's a base, it acts like a base. That means it can subtract a hydrogen. So as this is a basic enzyme, so they will subtract or abstract this, they will abstract this proton from this keto structure. So when they will subtract this proton, how they will do that? They will try to form a bond with this hydrogen. Okay, so the as the bond will be formed here, that means this bond will be, this bond will breaks down, and the electron which was shared here, they will come to here, form a transition double bond. Eventually a fully developed double bond will be formed here, and this hydrogen will get released from the keto structure, and due to that a transition negative charge will be formed here. That means the oxygen here, it will be negatively charged. So as they are negatively charged, then they can easily bind with a free proton. Okay, and thus enol will be formed. Okay, so as in this reaction, the enzyme is acting as a base. That's why it is called base catalysis. Okay, so this was the acid base catalysis, example of acid base catalysis. However, although this reaction show that the acid catalysis or base catalysis, they are occurring separately, but there are some reactions where both this mechanism can occur simultaneously. Okay, now covalent catalysis, another mechanism of catalysis. Okay, so in case of how the covalent catalysis occurs, it involves rate acceleration through the transient formation of a catalyzed substrate covalent bond. That means in this mechanism, the enzyme, they form a covalent bond with the substrate. Okay, and the whole process of covalent catalysis can be conceptually decomposed into three stages. Okay, first, the nucleophilic reaction between the catalyst and the substrate to form a covalent bond. Then the withdrawal of electrons from the reaction center by the now electrophilic catalyst, and then finally the elimination of the catalyst, and a reaction that is essentially the reverse of stage 1. Okay, let's try to understand these stages by a example. So this is an example. So this is the uncatalyzed reaction, acetoacetate, they turned into acetone, and in between they are in a transient form, enolate. Okay, acetoacetate, enolate, acetone. Okay, so catalysis who can form a catalyst who can form a covalent bond with acetate, they can facilitate these reactions to occur faster. How? Suppose this is a part of the enzyme RNase2. Okay, and this enzyme is a, and this part of the enzyme, that RNase2, can act as a nucleophile. That means they can attract nucleus. Okay, so as they are nucleophile, that means they have extra electron, because if a molecule wants to attract the nucleus, we know that nucleus is positively charged, so it need extra electron, so it has extra electron. Okay, so with the help of this free electron, they form a covalent bond with the acetate state, that is with the substrate. Okay, so you can see the RNase2, the NH2 group of the enzyme, they formed a covalent bond with the carbon here. Okay, so once it is done, so as it this bond formation occurs in by nucleophilic reaction. Okay, so that means the nitrogen, it has shared its extra electron with the carbon of the acetate. So as they have shared their electron, that means now they lack electron, so they become electrophiles. Okay, so electrophiles means they can attract electron. How they can do that? If they lack, that means the electrophiles, they lack electron, so that's why they can attract electron. Okay, so first it has extra electron, so with that electron, it has free electron. With that electron, it has formed the covalent bond with the carbon of the acetate, and now it has become electrophile. Okay, so as it has become electrophile, what it can do now it can attract the electron of this double bond. That means it can withdraw this electron from this electron center. So when it do that, due to this one, when nitrogen will withdraw the electron from this reaction center, because of the effects of this part, this bond will break down, and because of this, the carbon dioxide will get released, and the molecule will become like this, which is a transition state. Okay, so carbon, nitrogen, it has some uh, extra electron now, because it has abstracted or withdrawn this electron from the reaction center. Okay, and this bond was broken and the carbon dioxide was released. Okay, so after this step, this bound enzyme, it will get released from this transient structure. So how it will happen? This electron, okay, this electron will get back to its original position. Okay, and thus a double bond will be formed, and in this case a hydrogen molecule, it comes here, so a double bond will be formed here, and because of the formation of this double bond, will be broken, and electron will be now the electron will come close to the carbon, and it will become transiently negatively charged. Okay, so because of that, now it can bind with a proton ion, and this transient structure will be formed, and eventually hydroxyl ion, from the reaction environment, will react with this uh, part of the enzyme, and they will get detached from this structure, and that means the enzyme is now released from the structure, and they will become acetone. Okay, so this is the details mechanism of covalent catalysis, but if we want to say in brief, we call it covalent catalysis because the enzyme covalently bind with the substrate in this mechanism. Okay, so now in case of covalent catalysis, okay, there are both electrophilic stage and neutrophilic stage. Okay, so that means both nucleophilic catalysis and electrophilic catalysis occurs here. So which one is the rate limiting? We can name the reaction. So for example, the nucleophilic stage is rate determining step, then we can call it nucleophilic catalysis mechanism. Or if the electrophilic catalysis stage is rate determining, then we can call it electrophilic catalysis. Okay, now to some extent nucleophilic catalysis resembles that of general base catalysis. Okay, except that instead of abstracting the proton from the substrate, the catalyst nucleophilically attacks it to form a covalent bond. I mean, the base catalyst, they abstract a proton. Okay, but the nucleophilic catalyst, they don't abstract a proton, rather they form a covalent bond with the abstract by nucleophilic interaction. Okay, so if covalent bond formation is the rate determining step of a covalently catalyzed reaction, the reaction rate tends to increase with the covalent catalyst basicity. The more basic the catalyst is, the reaction will be more faster. Okay, so a good covalent catalyst must combine the properties of high nucleophilicity. Okay, so if it has high nucleophilicity, you can easily form the covalent bond with the substrate. Okay, and also the ability to form a good leaving group, that is to easily reverse the bond formation step. Okay, uh, one example of such catalyst is groups with high polarizabilities. Okay, that means with highly mobile electrons such as imidazole and thiol functions have these properties and hence make good covalent catalyst. I mean, the catalyst who have this group imidazole and thiol, they are good covalent catalyst. Okay. Okay. Okay, so we learned about the acid-base catalysis and covalent catalysis. In the next uh, part of this lecture, we'll learn about the other mechanisms. Okay, so thank you.